This is proteins, lecture number 3. We're going to talk about peptides. So we finished talking about amino acids up to this point. You know what form they exist in now, and you also know how to synthesize them, both in racemic fashion and using catalysts. You could make a specific amino acid as well. So we're ready to talk about what happens when these two amino acids get together, and what it forms is what we call peptide. And peptide is basically an amide formed from two or more amino acids. So that if two amino acids got together and formed an amide bond in between, and you made, then you would make a dipeptide. And if you had 3 residues or 3 amino acids put together with amino acids, and they would be called tripeptide, etc., etc. So depending on how many monomers are used in order to make the peptide, you could say, you know, how many are in that peptide. And once you get to be a big number, then you call it a protein. So we are going to start from very simple dipeptide where 2 amino acids get together. And so here's an example of an alanine and a glycine, 2 amino acids. And what has to happen is enzymatically, this is what will happen, is the carboxylic acid of one amino acid and an amine group of the other amino acid would have to come together to form this amide bond. So here is the bond that is formed. So this bond right here that I'm highlighting is the bond that is formed between the 2 amino acids. So this is the amide linkage that's responsible for keeping 2 amino acids covalently together. So this is alanine attached to glycine. Abbreviation would be alanine dash with glycine. So the question is this alanine glycine same as if I made dipeptide in which I have glycine first and then alanine? At first glance it seems like they should be similar, right? But what is the relationship between these two? Are they exactly the same? Are they two totally different compounds, structural isomers, or are they mirror image of each other? Let's take a look. All right, so here's from your book. We have an alanine and a glycine, and we made this bond right here. And then we put the glycine on the left-hand side so that the amino group of glycine is now free. And so I'm using the carboxylic acid of glycine with the amino group of alanine. And then make an amide bond. So the amide bond will be over here. All right. So now compare the two structures. What would you say is the relationship between these two compounds? Are they mirror image of each other, for example? And your answer should be no, they're not mirror images of each other. What are they then? They have same exact number of carbons and hydrogens and oxygens and nitrogens. So molecular formula is same, but they're put together totally differently, correct? So this would be an example of a structural isomer. And notice, depending on which amino acid is on the left side versus which amino acid is on the right side, you would be using the different functional groups, all right? So notice the amino acid on the left will use the carboxylic acid, and the amino acid that is on the right will use the amine to combine to make an amide. So this is how it's written in portraying peptides. Conventionally, you always put the amino acid that has a free amine group on the left side, and then you write your amino acid sequence, and then on the right hand side would be the free carboxylic acid. So this is sort of like, you know, you always write from left to right when you write your name, a word. So that's how it is with writing out peptides. You would start with the what is called N-terminal residue. So the amino acid that contains a free amine is the N-terminus, and then you write the amino acids, and then the amino acid that has the carboxylic acid free is called the C-terminal residue, right? So you have to obey this convention, because if you don't, then you're really not drawing or portraying the sequence of proteins in a correct way. All right. So now let's take a look at the amide bond that is responsible for linking these amino acids together. All right. So the amide bond that's responsible will look like this, just like the amide bonds that we've seen before. And what you remember from the acid derivatives unit is that this nitrogen lone pair is contributing in this way. So you could draw a resonance structure like this. And what that's telling us is that the amide bond, this carbon nitrogen, that has a double bond character. And that's a really important concept to keep in mind, because that sort of determines the conformation that peptides have. All right. So if you look at the typical peptide bond, and so here's one of them, what you'll notice is that the bond length, this CN bond length is at 1.32 angstroms, but on the other side where there's no resonance, this bond length is 1.47 angstroms. So that bond length already is telling us that there is a double bond character in that bond. Okay, so what are the consequences of this double bond character? All right, one consequence is that the nitrogen with the lone pair of amide is not nucleophilic nor basic. All right, and we've went over this before, so this should be on review for you. So we're talking about the reactivity of that nitrogen because it's busy sharing its electron density with the oxygen here, as shown here. The lone pair is really not 100% there, but it shares. So it's not going to be acting as a nucleophile or a base in amides. All right, so that's very, very different from an amine that's not a part of an amide. So like a totally isolated amine without the C double O would be basic and nucleophilic, but this is not the case for amides, obviously, right? And we already talked about the shorter the NC bond length, right? Because of the double bond character, the other consequence is that there is restriction about the CN bond, right? Which CN bond? The shorter one where there's a double bond character, all right? That also means that the atoms attached to the C double O and the nitrogen are in one plane. Okay, and that's what is shown here. Notice the nitrogen, carbon, and oxygen. Anything that is attached to them. So, for example, this hydrogen, this carbon, this oxygen. Basically, think of this as a C double C. You know that the atoms that are attached to C double C have to be in one plane. Same here. So these atoms are going to be in one plane, and so that causes a little bit of rigidness about it. All right. Now the other consequence is that because there's a double bond character, there's a way in which you could rotate this, and this R group here and this R group here. Notice if you pretend that there's a double bond here, this R group and this group are in a trans-like conformation, right? Whereas if I rotate it over and rotate it this way, actually, in this is sort of like a cis-like about this double bond, correct? So notice in the cis conformation that these carbons with the R groups are pretty close to each other, whereas when it's like trans-like, these groups are farther apart. So which one do you think is going to be more stable because it has less steric interaction? So obviously the trans-like conformation is better. Due to less steric hindrance. Right? So that is important in predicting what the peptide would look like in a 3D shape. All right. Now the other thing that sort of you have to kind of notice it is that not only is it in a trans fashion, but the side chains, the R groups for one amino acid and the side chain of the other residue, notice that they're in an opposite plane. Like this is above the plane of this amide plane, whereas the other side chain is below the plane. Okay, so that further reduces the steric interaction that it might have if it was both on the same side of the plane. Okay, so everything is sort of optimized to give the most stable conformation when you look at an amide bond, and this is sort of repeated over and over again when you look at a long chain in a protein as well. Okay, now if you're really curious as to what this looks like physically, what I recommend is that you make a model like this and then take a look at it. So here's a C double bond O, and here's the nitrogen. This is where the double bond character is. So this bond is sort of fixed. I'm going to do it quickly. And then this atom, this oxygen, the hydrogen, that's in one plane. And notice that this R group is on the opposite of, this is on top, this is in the bottom. And also this, if you think of this as double bond, notice that this is in a trans fashion. Okay, so this is what a dipeptide would look like.